Let us continue.
Excuse me, Paula. It's a meeting.
Why not? Five 45 years. Okay, now we were trying to understand some experiments with reference to plasma membrane. Try to understand what I told you is the concept of plasma membrane. We try to understand this with reference to a plant cell. Now, what's happening is, I said, we took a solution here. We don't know what is the strength of the solution. What do you understand now by the word strength? Whether the solution is dilute or the solution is concentrated. We don't know. But we are asked to add some cells here. We have taken some cells. Now, when we are talking about this, what I said, we are adding some cells here. What to add? I take some cells. What type of cells I am taking? Plant cells. I am taking some plant cells here. In all the three beakers, I am placing some plant cells. In all the three beakers, I am placing some plant cells, same plant. In all the three, I am taking some plant cells. एक ही plant के cells लेने हैं. I place them. So can we take the individual cells like this? No. What are we to add? एक plant part हम actually add करते हैं. But now, to make it clear for you, I am drawing a single cell. वैसे तो we cannot take a single cell in the beaker this way. So actually, what am I taking? Either a stem or a leaf. That is what we are putting in the solution. But to make you understand, we draw cells like this. So that you understand what is happening to a single cell in that leaf or in the stem. Are I समझ में? So now what I said, in three beakers separately, we have taken certain solution. And then we place some plant parts. We are observing some cells. Now see there, they are also talking about plant cells. Are I समझ में? Plant cells, they are talking about adding certain cells in a solution. I can try with plant cells or with animal cells also. The upper group of cells are the RBCs of the animals. And the lower line of cells, what type of cells are they? They are the plant cells. Now, after putting the plant parts here, we wait for some time. For how much time? Let us wait almost for about 8 to 10 hours. We put this setup like this and then we wait almost for 8 to 10 hours. In 8 to 10 hours time, we are to check the observation. We are to check the observation. Now let us see what are the observations. In the first beaker, what will be the observation? In the first beaker, we saw that the cell is remaining exactly the same. There is no change in the plant part. Again, I tell you, we are trying to show the cells for ease of understanding. Actually, it is just a part of the plant that you take. But हम cell क्यों draw करते हैं? So that we are able to express nicely. We are able to understand nicely, sit properly. Straight and smart. That's not the way to sit. You keep on hanging and swinging in the class in the school also. Which school you belong to? Sam. Now, this particular cell that I talk about, we are trying to understand, are there any differences in the cell shape, structure? अब जो पहले वाले भीतर में, I get to see no, the cells are not showing any change in the shape, in the size, shape, कुछ भी change नहीं है. So I will write, what about the cells? The cells remain the same. The cells are remaining the same. There is no change in the shape of the cell at all. Okay. What about the second beaker? In the second beaker, after a period of time, I get to see that this cell, which was originally like this, now the cell has started swelling up quite a lot. अंदर का जो vacuole है वो काफी बड़ा हो चुका है. So either you understand this way, or the swelling can happen in many different ways. I may understand ये जो cell था, ये जो plant cell था, that has started swelling this way. ऐसे भी होता है, where you understand that the vacuole has become much bigger than what it was earlier. How are we supposed to write? The cell became swollen. ये मैं क्या लिख रही हूँ? What I am observing after this time. आ रहा है समझ में? Third, talk about this. पहले ऐसे था, normal cells. अब कैसे? After 8 hours, now what? After 8 hours, I get to see that ये जो cell है, this cell has started showing some constrictions, and जो vacuole था, the vacuole has become much constricted. So I will write the cell has shrunken. The cell has shrunken. So now we try understanding that what is the reason for this? What is the reason for this? Now the reason for this would be when I said that this cell remained the same, matlab experimental period of time. Before the experimental period of time, the cell was this way. After 8 hours, what did I say? After 8 hours also, the cell is almost the same. After 8 hours also, the cell is almost the same. कुछ ज़्यादा change है? No. Is there any change? No. Much change है ही नहीं. The cell is almost the same. So we are writing the cell is same. Let us take the second condition में. Okay, the second condition is something like this. ये जो cell है, after 8 hours, what is the next condition? The cell has become swollen. The cell has become swollen. पहले से काफी बड़ा हो गया है cell. So what must have been the reason? The reason primarily is adequate amount of water has entered. Adequate amount of water has entered. Now, why did water enter? Because perhaps this is a dilute medium, dilute solution. And जो ये cell के अंदर, the plant cell has a big vacuole inside. And if I have to draw any plant cell, हम कैसे draw करते हैं? A big plant cell and then a vacuole. And the liquid inside the vacuole, what is the liquid inside the vacuole called? It is called sap. What is the meaning of the word sap? What does sap consist of? The sap has water and lots of dissolved salts and minerals. So I already told you that when I talk about a solution का मतलब क्या है? Water में salts dissolve रहना, iron dissolve रहना. So I can simply talk about salt solution मतलब where the water has a adequate amount of solute in it. That may be sugar, that may be salt, that may be ions. अच्छा, very simple. एक plant cell में जो vacuole रहता है, the vacuole is always filled with water. And it is not only water, but water में काफी सारे salts and ions dissolved. तो उसको हम क्या बोलेंगे? सैप. Now what am I doing? I am comparing this solution with the sap inside. And I get to see ये जो सेल जो स्वेल हो रहा है only because water is entered, इसीलिए. तो water क्यों enter होगा? Because this is a dilute solution and inside the sap it is a concentrated solution. I am comparing the sap of the cell and the solution outside the cell. Okay? And now I see that the water is entering from outside to inside. क्यों? क्योंकि यहाँ पे it is dilute. What did I say? Dilute मतलब 2 spoons of sugar and 100 ml of water. Concentrated मतलब 5 spoons of sugar and same water, 100 ml water. तो यहाँ पे water ज़्यादा है and the water is moving from a region of high concentration to a region of low concentration. So first of all we learn the definition of the word osmosis. Write down. What is osmosis? पहले हम ये सीखेंगे. What is osmosis? The movement of the water from a region of its high concentration to a region of its low concentration through a semi-permeable membrane. Do you remember the meaning of the word semi-permeable now? Selectively permeable, semi-permeable किसे बोलते हैं? A membrane which will allow the movement of some molecules only. ठीक है? A membrane which will allow the movement of only some molecules, not all. उसको बोलेंगे semi-permeable. So now, in the second condition, we see that the cell is swelling up. The cell is swelling up. So in the next condition, the second condition what I showed you, first condition cell remains as it is. In the second condition the cell is swelling up. Why? Because water is entering the cell. Can we say that this is osmosis? When we are talking about that the water is entering, again come to this figure. What is happening here? Water is entering from the beaker to the cell. Follow the definition now. Water is moving from a region of high concentration to a region of low concentration. I repeat, water is moving from a region of high concentration to a region of low concentration. Is that correct? So when a cell gets water from outside, when the water is entering the cell, what type of osmosis this is? Endoosmosis. We try to write this. Two types of osmosis. Two types of osmosis. What are the two types? First है endoosmosis. What is the meaning of this? When water enters into the cell. When water enters into the cell, this is endoosmosis. When water enters into the cell, or ये आया समझ में? Let us write the definition. I said there are two types of osmosis. What are the two types? एक है endoosmosis. When the cell is taking water, when the water is entering the cell, उसको बोलेंगे endoosmosis. But the question is, when should water enter the cell? When will the water enter the cell? When outside the cell, the solution is outside the cell, solution is dilute. यहाँ पे water ज़्यादा होगा ना? The rule of osmosis is what? What is the definition of osmosis?
When water enters the cell.
When movement of water takes place from a region of high concentration to a region of low concentration. Again I repeat, this is a dilute medium, this is a concentrated, इसीलिए water enter हो रहा है. The rule of osmosis always remember, water from a high region moves to a region of low concentration. So when the water enters the cell, what will be the nature of the cell? What will happen to that cell? The cell will swell up. What is the reason for the swelling? Your answer is osmosis, preferably endoosmosis to write. अच्छा, ये हो गया सेकेंड. अभी थर्ड कंडीशन में वही आफ्टर 8 hours के बाद we get to see that ये जो सेल है, the plant cell has started shrinking. The plant cell has started shrinking. ये था थर्ड ऑब्जर्वेशन. तो shrinking क्यों होता होगा? Only because the plant cells must have given away so much of water. Cell sap में जितना water था, काफी सारा water बाहर निकल गया है. Something like when the grapes, you have seen the grapes so fleshy and so full of water. But when they get converted or when they are dried, raisins बन जाता है ना? Raisins are so dried up. But उसी raisin को फिर से हम पानी में डालते हैं तो they swell up again. नहीं? किशमिश को जब हम पानी में रखते हैं, they swell up again, no? Raisins को जब हम पानी में रखते हैं, they swell up again. Why do they swell up? Outside there is so much water, the water enters the raisins and they start swelling. But grapes को with using some preservatives and all, अगर grapes को हम सुखाएंगे under the sun or heat, they will start drying out. क्यों dry? Because they are giving away water. So these are the two opposite things we are trying to learn now. Why does a cell swell up? Only when they take water. What is this process of taking in water known as? Endoosmosis. Why does a cell shrink? Because it's giving away water. What is this process of giving away water known as? Exosmosis. This is known as exosmosis. The process of giving away water is known as exosmosis. अभी ये बताओ, which one is a dilute environment? This or this? This water in solution in the beaker is dilute, or sap solution is dilute. Which one is dilute? Sap is dilute. Meaning, if I say यहाँ पे solute ज़्यादा होगा, salt, sugar जो भी है, compared to what is there inside the sap. इसीलिए the water is moving from a region of high concentration. Very good. From a region of high concentration to a region of low concentration. Osmosis का definition हमेशा याद रखना है. What is the definition of osmosis? Movement of water from a region of high concentration to a region of low concentration through a semi-permeable. तो इस सेल में semi-permeable membrane कौन सा है? ये जो सेल में I'm saying water is going out, हाँ, and water is coming in. यहाँ पे water बाहर जा रहा है, यहाँ पे water अंदर आ रहा है. Semi-permeable membrane कौन सा है? Cell membrane. ये जो cell membrane है, this is the semi-permeable membrane, nuclear membrane. क्यों है हो ना? Nuclear membrane नहीं, cell membrane. We already learned, no? Were you there in the class last period में?
This is my first lecture.
The last period.
Last period. Understand? Yes. Are you here in this class? कि कहीं और मैथ्स चल रहा है मुझे पहुंचना है वहां पे ऐसा है नहीं ना तो उसका गुड मॉर्निंग अभी हुआ है गुड मॉर्निंग हो गया तुम्हारा हाँ now are you clear with these two definitions what is the meaning of a dilute solution what is the meaning of a concentrated solution तो डाइल्यूट सॉल्यूशन में सॉल्यूट ज्यादा रहता है कि कॉन्सेंट्रेटेड सॉल्यूशन में सॉल्यूट ज्यादा
Dilute.
concentrated solution अगर remember I had explained you with 100 ml of water दोनों ग्लास में 100 ml of water है एक में मैंने 4 spoons of salt लिया दूसरे में मैंने 1 spoon of salt लिया कौन सा डाइल्यूट है 1 spoon of salt same 2 glasses of water always to remember what is the meaning of dilute and what is the meaning of concentrated if I talk about 1 teaspoon of salt and 100 ml water if this is I'm sorry 4 teaspoon of salt and 100 ml of water so dilute I will say 1 teaspoon plus 100 ml of water so ये comparison याद रखना है once you are clear about this then it is easy to understand these two concepts why does a cell shrink because the cell gives away water what is the process of giving away water called
Osmosis.
what type of osmosis
Exosmosis.
exosmosis ये हमेशा उल्टा क्यों बोलते हैं the process of giving away water what is it called exos and what is the process of taking in water called endoosmosis but is it not the definition should remain the same or not let us check I am saying this is endoosmosis ठीक है ना अब यहाँ पे क्या हो रहा है the water is moving from a region of high concentration to a region of low concentration ठीक है क्योंकि ये dilute medium है ये concentrated है let us check यहाँ पे we see that the cells are shrinking हाँ पर यहाँ पे भी the water is moving from a region of high concentration to a region of low concentration अब ये दो solution में when I try to understand movement of water from a region of high to low ये समझो I am saying in this solution there is more solute in this solution there is less solute अच्छा if I say something around 200 ml of water 200 ml of water and यहाँ पे 1 teaspoon and 100 ml of water which one is dilute which one is concentrated second one is still concentrated second one is still concentrated the amount of water is double solute is not double no it is more than double तो ये फिर भी अभी भी concentrated ही है so that way you have to understand concentrated and dilute का क्या मतलब है exosmosis and endoosmosis this is the definition of endoosmosis can you write the definition of exosmosis on your own now try writing down the definition of exosmosis आप कौन सा change करना है when water enters नहीं leaves the cell when water leaves the cell because the sap is dilute as compared to the surrounding medium because the sap is dilute as compared to the surrounding medium because learn two more words. What are those? Dilute solution ko we can also define the word dilutum. Dilute solutions as hypotonic solution. Dilute solutions are also known as hypotonic solution. Concentrated solutions are also known as hypertonic solutions. Let us learn these two terms. Let us compare here. I am saying in this cell, water was entering. In this cell, water is entering. Let us talk about this condition. This is a cell. Water is entering. Why is water entering? Because outside is dilute and inside the cell is concentrated. So the solution inside the cell is what we are terming. This solution inside the cell, now we will say the cell is hypertonic as compared to the outside medium. The cell sap or the liquid inside the cell is concentrated. What is the other word for concentrated? Hypertonic. So I am saying inside the cell solution is hypertonic. Outside is hypotonic, dilute hai. So agar inside the cell it is hypertonic medium, if the inside is hypertonic concentrated, then only the cell is gaining water, receiving water. आया समझ में? अच्छा चलो ये देखेंगे. What is happening? The cell is shrinking, no? The cell is becoming small. Why is the cell becoming small? Because the cell is giving away water. Which medium? This or this is concentrated. This part has more solute, less water. Concentrated. This is a concentrated solution. This has less solute, more water. Dilute solution it is. That is the reason this cell is giving away water, because it has more water than the surrounding. Giving away water, giving away water, giving away water. Process exosmosis. What is this process of giving away water? Exosmosis. Understood? So now, can I say that this cell is hypotonic as compared to surrounding? Is my statement correct? This cell is hypotonic. Hypotonic means dilute. इसीलिए तो वाटर दे पा रहा है. अगर अंदर concentrated होता तो it would have drawn water inside. जैसे ऐसे. अभी the cell is not losing also much, gaining also much. मतलब something like the first condition. तो ये कैसे? What is happening in this condition? क्यों? After 8 hours also the cell is exactly the same. Because जो एक दो molecule अंदर आया भी होगा, if one or two molecules must have come out, another one or two molecules must have got in also. ये remarkably हम endosmosis भी नहीं बोल सकते हैं, exosmosis भी नहीं बोल सकते हैं, because अगर endosmosis होगा तो regularly endosmosis होते होते सेल बड़ा होने लगेगा. Exosmosis होगा तो सेल will keep shrinking. But हाँ ये दोनों चीज यहाँ पे applicable नहीं है. तो क्या रीजन है इसका? रीजन यही है, the outer environment here and inside is more or less the same. Something like, if I say in simple words, शायद यहाँ पे अगर 1 teaspoon of sugar and something like 100 ml of water add किया है, तो यहाँ का concentration भी लगभग same ही है. Concentration here and the concentration here is almost the same. इसीलिए by chance अगर 2-3 molecule अंदर भी गया है, another 2-3 molecules will also be simply come out. But कोई forcible entry भी नहीं हो रहा है and forcible exit भी नहीं हो रहा है. Like endosmosis and exosmosis. तो ये वाले solution को, this solution which is same as the cell, this type of solution is isotonic. Iso, same. Isotonic. Now, actually, we are defining the solution outside the cell. Let me again tell you. I am saying this solution outside, let us define with reference to the solution outside now. Try to understand. This solution is isotonic. That is the reason if few molecules go, few molecules come out. Ultimately, there is no remarkable change in the cell. So if I put my question this way, when the cell does not suffer from any change, when the cell is placed in a isotonic solution. आया समझ में? अच्छा, अभी यहाँ पे the cell is receiving water. What type of solution is this? Hypotonic. No? This is dilute. यहाँ पे water ज़्यादा है, तभी water enter हो रहा है. High concentration to low concentration. High concentration to low concentration. So इस solution या outside solution को हम क्या बोलेंगे? This is hypotonic. यहाँ पे what type of a solution is this? ये बाहर वाला solution. What is this? Hyper. तभी तो this is hyper. This is hypo. Hypo से hyper हमेशा water move होता है. Direction of water movement is always from dilute to concentrated. Hypo to hyper. और अंदर सेल के अंदर और बाहर अगर concentration same रहेगा, then जब सेल के अंदर और सेल के बाहर concentration same रहता है, isotonic. At that time, is there any major movement of cell water molecules? No. At that time, there is no major movement of the water molecules. So now, what are the three conditions? When the cell is placed in an isotonic solution, the cell remains the same. When the cell is placed in a hypotonic solution, the cell swells up. When the cell is placed in a hypertonic solution, the cell shrinks and may die out also if kept here for a long time. Let us write down this definition. 1, 2, and 3. Very, very important. Number 1, because isotonic solution. Isotonic. Spelling check करो और लिखो. Isotonic solution. A solution which has same concentration of solute. A solution which has same concentration of solute and water. Same concentration of solute and water inside the cell, as well as in its external environment. Number 2 लिखो, hypertonic. लिखो hypertonic, a solution which hyper PER, hypertonic, a solution which has higher concentration of solute, and lower concentration of water. Lower concentration of water as compared to a cell. As compared to a cell. As compared to a cell. Number three. Hypotonic solution. A solution which has higher concentration of water and a lower concentration of solute. Higher concentration of water and a lower concentration of solute as compared to a cell. आया समझ में? As compared, तो हमेशा कंपेयर करना है, एक मीडियम को दूसरे मीडियम से हम कंपेयर कर रहे हैं as compared to a cell. So this is what you have to learn about the plasma membrane and the concept of osmosis. Now we take up the plant cell. Take out your pencils and draw this diagram. Plant cells. करो 10 minutes में finish करो diagram. Not more than this. Whatever. Draw and neatly label all the parts.
And we have to also label as pencil.
Drawing and labeling all with pencil. You should not use any pen. When you draw the diagram, label the names. Label the parts all with pencil. Done? Okay. Why you need time? क्यों देखना है उसका डायग्राम क्यों देखना है तुमको? हाँ? Give it back. I have put one diagram on the screen there. I have drawn the diagram on the board. फिर से तुमको उसका book क्या देखना है? Is this two diagram not enough to make you understand how you are supposed to draw? I need to judge this diagram how you have drawn. Fatafat, last couple of minutes, finish up. Done, all of you? Now, we are going to pick up some important parts of the plant cell to understand the detailed structure. It has cell wall that you have to study, plastids that you have to study, and the vacuum. Three important parts of the plants. Very basic concept about the structure and function we are going to study. Let us start with the cell wall. Cell wall. What are some of the properties of the cell wall that we should know? Structure, function. Cell wall composed of cellulose. Cell wall composed of cellulose. The cell wall is rigid. The cell wall is rigid. Third property of the cell wall is the cell wall is freely permeable. Cell wall is freely permeable. What is the meaning of freely permeable? Semi-permeable, you know now. Semi-permeable means selectively permeable means
They choose this cells, they let some cells enter and
some molecules enter a membrane which allows the movement of only some molecules and does not allow the movement of another few molecules is a selectively permeable membrane. So if that is the definition of selectively permeable, you understand what is the meaning of freely permeable? A membrane which allows the movement of all molecules in, all molecules out. Write down, cell wall is freely permeable. Write down the definition of freely permeable. A membrane which allows, a membrane which allows, a membrane which allows.
And do we have to write all that or
Yes, yes. ये 3 पॉइंट लिखो और उसके बाद से फ्रीली परमेबल के नीचे लिखना है. देखो फटाफट पहले 3 पॉइंट्स लिखो उसके बाद थर्ड पॉइंट पे दिस एक्सप्लेनेशन यू हैव टू राइट. अभी तुमको नींद नहीं आ रहा है? हाँ, क्या नाम है तुम्हारा? आर्यन. A membrane that allows the movement of all molecules. A membrane that allows movement of all molecules through itself. Movement of all molecules through itself is known as a freely permeable membrane. Is known as a freely permeable membrane.
Can you repeat?
A membrane which allows the movement of all molecules through itself. Through itself is called, is known as a freely permeable membrane. Full stop. Sometimes you get questions. Explain, cell wall is freely permeable. Explain, cell wall is freely permeable. अच्छा, तो ये हो गया cell wall is freely permeable, cell wall is rigid, cell wall is composed of cellulose. Now we'll try to understand what are some functions of the cell wall. Functions are more or less like the cell membrane that we have studied. So functions of cell wall: 1, holds or gives rigidity. Gives rigidity to the cell. gives rigidity to the cell. Prevents bursting of the cell, especially in aquatic plants.
aquatic plant means the plant that ocean.
वाटर में जो रहता है, वाटर प्लांट्स, उसको बोलेंगे aquatic plants. Can you give me one example of aquatic plant? Kelp is a weed. वो plant कहाँ है? Aquatic plant. Hydro plant नहीं है. Aquatic plant नहीं देखा, water plants नहीं देखा कभी. नाम नहीं पता. हाँ? Coral plant नहीं है. Lotus. ये तो सबने देखा है ना? हाँ? Shoe flower कहाँ पे water में है? Shoe flower or hibiscus is the flower you get to see on the land. That is the land plant. There is still plant that is. Aquatic है, something like hydrilla. Hydrilla. Something like very common if I have to name hydrilla है, water hyacinth है. मतलब these are plants जो तुम near the water body हमें मिलता है. या तो even deep in the water body भी present हो सकता है. But very common if I talk about hydrilla and all, near the lake, pond, sea, if you go near the sea also you get to see some plants on the seashore. Hydrilla, very common. And lotus everywhere you get, no, on the ponds and lakes, lotus is बहुत common in this region especially. So ये हो गया cell wall. Now we try to study the plastids, a very important cell organelle for the plant. Plastids. Now we talk about the plastids, the second cell organelle. Plastids. Plastids. The first structural concept about plastids consists of double wall. Number 2, possesses stack-like structures known as grana. Cytoplasm is known as stroma. अच्छा, let me show what is grana stroma. ये जो plastid है, अगर if I want to draw a little bit big, कैसे draw करूँ? It is something this way, which has a large number of plate-like structures. इस तरह से plate-like structures, plate-like structures, plate-like structures, plate-like structures, and these plate-like structures they are connected. तो ये जो group of plate-like structures हुआ, क्या हुआ? Mosquitoes. This is known as grana. स्टैक को जैसे सपोज हमारे घर में प्लेट्स एक के ऊपर हम रखते हैं ना स्टैक करके बिना यहाँ पे हम रखेंगे कटलरी सो वी राइट स्टैक सो इट इज समथिंग लाइक दैट. तो वो पूरा स्टैक ऑफ प्लेट्स को बोलेंगे ग्राना. और ये जो साइटोप्लाज्म है इसको बोलेंगे स्ट्रोमा. स्ट्रोमा एक प्रॉपर्टी जो प्लास्टिक में है जो बाकी सारे ऑर्गेनल्स में नहीं है is it has its own DNA. Plastids has its own DNA. एक और ऑर्गेनल हमने सीखा था which has its own DNA. कौन बोले? तुमने भी डिस्कस कर दिया. राइट के हीरो बोलो, बताए आंसर.
Nucleus.
अभी मम्मी को फोन लगा. Chromosomal chromatin. I said, I am talking about a cell organelle ऑर्गेनल जिसके पास अपना खुद का डीएनए है। वैसे तो डीएनए हम सीखते हैं न्यूक्लियस में है। ये प्लास्टिड एक सेल ऑर्गेनल है जिसके अंदर डीएनए होता है। कौन सा और एक ऑर्गेनल हमने पढ़ा था? और कौन सा एक और ऑर्गेनल हमने पढ़ा था जिसके अंदर अपना खुद का डीएनए रहता है? हाँ, आज पढ़ा था मैंने बॉडी के अंदर डीएनए है। एक केबीसी खेल रहे हो तुम मेरे साथ।
मैम क्रोमोसोम्स
इसने बोला था माइटोकॉन्ड्रिया, two organelles which have their own DNA. Number one is mitochondria, number two is plastids. ये याद रखना है। Two organelles which have their own DNA. तो first है cytoplasm is known as stroma. और क्या लिखेंगे? So the function structure possesses its own DNA.
And you have not given that mitochondria as
नहीं दिया था?
नहीं।
हाँ तो लिखो वहाँ पे, लिख के रखो for mitochondria. Did you draw the diagram of mitochondria?
Yes,
वहाँ पे mitochondrial DNA नहीं लिखा?
नहीं।
डायग्राम में mitochondrial DNA नहीं लिखा?
Yes, ma'am.
हाँ तो लिखा था ना, तो वहाँ पे पॉइंट में भी लिख के रखो। I must have explained but पॉइंट तुमने नहीं लिखा होगा। I'll show you again structure of mitochondria. देखो written ना?
नहीं, yes, ma'am. Diagram में लिखा है।
देखो दिखाई दे रहा है mitochondria has got its own DNA. Another cell organelle which has got its DNA is plastids. So possesses its own DNA. That is the reason it is known as semi-autonomous. देखो, it possesses its own DNA. इसीलिए इस ऑर्गेनल को सेमी ऑटोनोमस कहा जाता है। व्हाट इज द मीनिंग ऑफ सेमी ऑटोनोमस? व्हाट इज द मीनिंग ऑफ ऑटोनोमस? जो इंडिपेंडेंट है, किसी पे डिपेंडेंट नहीं है, उसको बोलते हैं ऑटोनोमस। तो इसको सेमी ऑटोनोमस क्यों बोला जाता है? व्हाई माइटोकॉन्ड्रिया एंड प्लास्टिड्स आर नोन एज सेमी ऑटोनोमस? बिकॉज़ ये पार्टली न्यूक्लियस पे डिपेंडेंट है, फुल्ली न्यूक्लियस पे डिपेंडेंट नहीं है। हाँ 50-50 उसको आएगा। Today you are, for example, you all, are you semi-autonomous or autonomous? अरे semi-autonomous बोलो बेटा। Today you are semi-autonomous. तुम अपना काम भी कर रहे हो, but फिर भी पेरेंट्स पे डिपेंडेंट हो ना ज्यादा से ज्यादा। But after 10 years, you will be like us, autonomous. You have to do everything on your own. Every decision you have to take on your own. सब कुछ अपने आप करना होगा. And you'll be able to do that. Then we will say that we are autonomous. So that is the meaning. Then which are the organelles which are autonomous? Are there any organelle in the cell which is autonomous? No, there is no organelle in the cell which is autonomous. क्योंकि सारे के सारे organelle nucleus पे dependent है. खाली दो ऑर्गेनल है जो पार्टली न्यूक्लियस पे डिपेंडेंट है, क्योंकि उसके पास अपना खुद का डीएनए है। एंड व्हिच आर दोस टू ऑर्गेनल्स? माइटोकॉन्ड्रिया, प्लास्टिड्स। सेमी ऑटोनोमस। फंक्शंस लिखो। अच्छा पहले हम टाइप्स, ओके, फंक्शंस एंड टाइप्स टुगेदर हम सीखेंगे। फंक्शंस, plastids can be of three types. What is the first type? Chloroplast. Chloroplast, they are green in color. Chloroplast produces chlorophyll and helps in photosynthesis. Helps in photosynthesis. What is the next type? What is the next type? Chromoplast. Chromoplast produces colored pigments such as carotenoids, which give color to flowers, flower petals, and fruits. Or some vegetables. Can you give me one example of a colored fruit? Non-green. Orange, mango, papaya, and whatever. And crop, beetroot, carrot. So ये जो सारे parts of the plant है which are non-green is also because of a particular type of a pigment. Carotenoids. I have given you only one example, such as carotenoids, carotenoids. Carotenoids, the orange color pigment, red color pigment, होता है that the plant can produce. So such type of pigments are produced by chromoplast. And chloroplast, they produce chlorophyll. जिसकी वजह से leaves are green. And leaves can undergo the process of photosynthesis. Okay? So produces chlorophyll and helps in photosynthesis, a major function जो plants perform करता है. Produces chlorophyll, helps in photosynthesis. Chromoplast produces colored pigments. They are colored plastics. So यहाँ पे also to remember, chloroplast means they are colored plastics. Chromo. Chloro मतलब खाली green. And this is chromo. There is one more plastic that we have to learn. I'll write here. Number 3. Leucoplast. Colorless plastic this is. Colorless plastic. They don't produce any pigment. कोई तरह का pigment नहीं produce होता है. They are colorless. And these plastics are present in any or all types of plants. सब में present होता है. Happy swimming. Next is function क्या लिखेंगे? Stores starch or oil droplets. ठीक है? Helps in the storage of starch and oil droplets. Leucoplast. Leuko. Leucoplast. So what are the 3 types of plastics?
Chloroplast, chromoplast, and
Leucoplast. Functions याद रखना है. Chloroplast, chromoplast, leucoplast. The last organelle that we have to talk about is, what is the other organelle left? Vacuole. Let us learn about the vacuole. Now, वैसे तो vacuole, the parts very simple. Let us only learn one basic term with reference to vacuole. Vacuole, when we talk about, what are some of the features of vacuole? It is consisting of single wall. It has got a single membrane or a single wall. It has a single membrane, single wall. Vacuole. And next is what? What does it contain? What did I tell you? What is the name of the solution inside? Contains cell sap. Function क्या लिखेंगे of the vacuole? I erase this.
Yes, ma'am.
Leucoplast erase करो.
Yes, ma'am.
Now, function क्या लिखेंगे? What are the functions? stores water and minerals. Stores water and minerals. And number 2, gives shape to the plants. Gives shape and rigidity to the plant cells or the plant parts. Cells matlab parts are made up of cells, no?
And in the first function plant we write it shows cell sampling.
हाँ, cell sampling लिख सकते हो, but it's always better that you describe it. Stores water. Cell sap का मतलब क्या है? Because already you have written structure में it contains cell. तो cell sap का मतलब ये भी तुम लिख दो. Cell sap contains water and minerals. So this is all about the chapter cell. So we are done with the first chapter. Now we have to solve many questions. So questions I'll take up in the next class. Today we don't have much time to solve questions. Now what I want you to do is, in the volume, we have whatever I have covered, all this is given in the volume. Now first of all, before I move into the section question part, you have to read the text also. Now before we meet the next time, before we meet for the next lecture, what I want you to do is read the text. Your NCRT you have no at home. The book you are supposed to read properly. You are supposed to read the text properly. Then only you will be able to solve all. So text घर पे पढ़ोगे अच्छी तरह से. कहीं पे कुछ भी doubt है, you will come and tell me the next class. अभी at least let us start with some few questions. For all questions that we have to solve. Abhi all will not be able to complete, but only one question I want to complete, which is this one, page 92 में page fill in the blanks करो. Page 92 में only one set of questions अभी करो. The next class will solve many more. Page 92 में first question करो, fill in the blanks. Page 92. चलो फटाफट get started. Page 92, fill in the blanks, complete करो. क्या? Did she ask you something? Did she call you? No. Then you do your work, Risha. Chalo, you have your book now. Then what are you waiting for? Should I ring a bell to start the work? Now, complete karo. I'm going to ask you what you all have written. You should finish up. Fatafat. Pencil se likho. If there's a mistake, you can erase and write the correct answer. Confusion है? पूछो.
And the cell wall of the plant cell prevents bursting of cell and dash solution.
Hypotonic. Dilute है ना? Bursting of cell in hypotonic.
The cell wall is found in the cells of plant fungi and
Fungi.
fungi and the cell you have to decide.
Cell wall is found in the plant cells, in the cells of fungi, and also in the cells of bacteria. Also in the cells of bacteria.
Plastids found in most plant cells are of two types, known as chloroplast and
Chromoplast. What is it?
Chromoplast that contains dash are called chloroplast.
Chromoplasts that contain chlorophyll are called chloroplast.
And the functions of the Golgi apparatus are the dash modification and dash of the substance manufactured in the cell.
Secretion और synthesis वो फर्स्ट क्लास में आएगा. Synthesis आज मैंने वो सारे चीज एक्सप्लेन किया था. Synthesis है SYNTHESIS. Synthesis, modification and packaging.
The cells which do not have membrane bound organelles are called
Prokaryotic. Prokaryotic ये हमने फर्स्ट एक तो लेक्चर्स में सीखा था. Prokaryotic, eukaryotic. हो गया? All done?
The primary function of leucoplast is
Store starch.
store starch.
Storing of starch. Storing of starch.
And oil droplets.
And oil droplets. अगर एक फंक्शन पूछा है तो storing starch.
The primary function.
Primary storing starch. Main function. Done? Done? Did you finish? All done? Now, so homework, what is homework I gave you today? No written work today as of for homework. but you have to read the entire chapter, NCRT and the volume. Then next class when we meet, when I ask you to solve the questions, you will be in a better position to solve all the questions. Okay? And at the same time, if any doubts, you can ask doubts. That's all.
And what if the last and the method of plant cell helps to maintain the touch of the cell?
Shape of the cell. Shape of the cell. Okay? See you again in the next class. Hopefully we'll start with the next chapter, that is tissues, the next. Okay?
सब चालू है मेरे क्या